So this is the pre-lab lecture for Experiment 1 for Bio 236 lab, and it contains information pertaining to both the in-person lab as well as the online one. So there are some slightly different objectives for the in-person and online labs, but they are related to each other. And the overall theme of this kind of Experiment 1 is to get you familiar with how worms are grown in culture in the lab, start looking at worms, how we would do a basic lifespan assay, and then thinking about planning an experiment using C. elegans to study aging. And so in person, we're going to make some reagents, including NGM plates for experiments. We're going to look at C. elegans under the microscope, particularly younger and older worms, and observe some differences between them. I'm going to teach you how to maintain or transfer worms by picking. And then, rather than conduct a lifespan assay, which actually uses a lot of reagents and a lot of time, we're in person, we're going to do it online, because I think we can get a lot out of it in that way as well. And then you're also going to start working on your independent projects online. You're going to choose one of 4 compounds that have been shown to extend lifespan, and then find 2 journal articles about the effects of that compound on lifespan, either in C. elegans or in another organism, and summarize them. And finally, there's going to be a short assignment to complete, basically a calendar that will help you plan an aging experiment using C. elegans. And there's more information about all this stuff in the protocols that are actually posted online. And so I just wanted to take a minute and talk a little bit more about using C. elegans as a research organism, particularly to study aging. And so we've talked a little bit in lecture about the advantages of using model organisms, and C. elegans is kind of one of the best for aging studies. C. elegans is a nematode or a roundworm, and it is free living, so it can be found in nature, usually living in some rotting fruit or decomposing matter. And it reaches about 1 millimeter in length, so technically it's visible with the naked eye, but you really do need a microscope to see any sort of detail, like you can see in the worms here on the right-hand side. And C. elegans has a couple things that sort of make it really useful for studying aging, one of which is the fact that it has a short generation time. It can go from an egg to a fully grown adult in about 3 days. And C. elegans also has a relatively short average lifespan of about 20 days. And that means that you can basically do an aging experiment in less than a month. And so you can imagine that you can generate a lot of data a lot quicker, at least pertaining to aging, than you would if you were working with mice or with humans. Another advantageous thing about C. elegans is the fact that it has the transparent body. And so these worms are all imaged live over time, and so this is the youngest worm at the top and the oldest one at the bottom. And you can actually see both tissues, cells, organs, and what's basically going on inside of a live worm over time, which is really advantageous for looking at tissue degeneration or cellular changes that are happening with aging. And so, for example, these are the heads of the worm on the left and the tails on the right. And you can actually see up in this worm here, this bulb structure is the pharynx, so you can see a full muscle structure. These ovals on the bottom are all eggs or embryos, which you can see inside of the worm. The intestine is here on the top of this worm, and it becomes much more pronounced in this one. And everything starts to fall apart as the worm gets really old. But that transparentness is really cool for studying differences with aging. And another really awesome thing about C. elegans is that their whole genome is sequenced, and it's pretty well annotated or kind of marked up at this point. So you can actually use C. elegans to identify genes pretty easily that are involved in aging. There is what's known as an RNAi library, or a library of constructs that you can use to knock down expression of any gene in the worm that you want, and then basically see what happens. And so if you have a gene candidate that you might have identified in a human patient, and there's a homolog or a similar gene in the worm, you can use the RNAi to knock down that gene and see what happens in a living organism. So the power of C. elegans genetics is really strong and really helpful in research in general, but definitely helpful in aging research. And finally, one thing that's nice about working with worms is that you can create a genetically identical population really easily. C. elegans is a hermaphrodite, and so it does not require mating in order to be maintained in the lab, which makes it a lot easier because the worms sort of maintain themselves. And it also means that each progeny is genetically identical to the parents, right? So one worm is generating all of its babies from its own DNA. And that means that the population of worms, if you chose one single parent to put on that plate, would all be genetically identical. And so you can create a very large sample size and also sort of remove the effects of any genetic background differences between animals. And so we can sort of take away the genetic contributors to aging by looking at a single population of worms from one parent, and then actually start to study how the environmental effects really how the environment can really affect aging. And growing or culturing C. elegans in the lab is actually relatively simple and relatively cheap. And so they can be grown in a liquid medium, like a broth, or a solid medium, like you can see here on the right, on basically plates or petri dishes filled with this media. And the media that C. elegans grows on is known as nematode growth medium, or NGM. And so they spend their lives on these petri dishes filled with NGM agar, and they eat a bacterial food source. It's a non-pathogenic strain of E. coli, which is known as OP50. And so OP50 is also relatively slow growing. You can actually see a lawn or kind of a spot of it here on this NGM plate. And that's advantageous because growth media in general are very nutritious. And so it's good to be nutritious for the worms, but it's not good to be nutritious for bacteria that are growing on the same plate. And OP50 is an E. coli that actually grows pretty slowly. And so it does not take over the plates. It allows the worms to still thrive and have a food source there. And so as the worms live, and they're hermaphrodites, right? So their population is expanding, and then all of the babies are also eating this food, you need to move worms from a plate that is depleted of food to one that actually has food on it, or a new lawn of OP50 bacteria. And so the way that you do that is using this instrument here, a worm pick, and you basically pick worms under the microscope from this plate to a new freshly kind of filled plate of bacteria, and that happens about every 3 to 4 days in order to keep a healthy culture of C. elegans going in the lab. And I'm going to teach you how to make these plates, grow up this bacteria, and put it onto your NGM plates so that you have a source of food for the worms, and also how to transfer them from plate to plate using a pick like this. And so C. elegans' life cycle has been well documented, and their development at specific temperatures has also been well documented, and it's highly, highly predictable. So the time of each developmental stage is highly predictable. The size of the worm at each developmental stage is highly predictable. And so you can actually plan experiments by calculating the number of hours after egg laying. And you can pretty much count on those developmental stages to be accurate. And so C. elegans is laid out of the reproductive adults as an egg or an embryo, and then it goes through 4 larval stages before it becomes a reproductive adult. And those larval stages are called L1, L2, L3, and L4. And then after L4, the worms enter reproductive adulthood. You can see actual micrograph images of that down here. And you can see the size changes between the different stages. with L1s being really small as the first larval stage, and L4s being much larger, and then adults being obviously even much larger than that. And then after reproductive adulthood is achieved, we can kind of start to study aging. And so that means at 20 degrees, which is the temperature we normally keep C. elegans at about 65 hours post egg laying, or after the egg is laid out of the worm, we can then kind of start studying that aging, right? Because it takes about 10 hours for that egg to hatch, and then another 16 hours before it moves from L1 to L2, and then by 65 hours, all the worms have reached reproductive adulthood, and anything after that is considered aging. So in order to measure the lifespan of a C. elegans, you have to start with a synchronized population of worms, or worms that are all at the same developmental stage. And you will choose a specific number of worms, somewhere around 100 usually, and put them onto a plate. And you can choose different stages at which to synchronize worms. In this class, we're going to use eggs or embryos to synchronize our populations of worms, but you can also use that larval stage 4 or L4 to do it. It's just a little bit more difficult to identify, and so we're going to use eggs for this class. And after you synchronize your population, you basically just have to wait for those worms to reach adulthood, right? And so if you're synchronizing at egg, you wait 65 hours, and then you have an adult population, which you would consider day 1 adults. And then every day after your worms reach adulthood, you will score or count the number of worms that are alive, and then transfer those live worms to a new plate. And this happens, you can do it every day, every 1 to 2 days. And this process will continue until you score 0 worms alive on your plate. And then the whole thing is graphed as % survival versus age in days. Right? And so it's a pretty simple process, right, of just looking at a plate of worms and scoring how many are alive, moving the live worms to a new plate, and then coming back the next day and doing it again until all the worms are dead. And what you get is a graph that looks something like this, with % survival here on the y-axis and age in days on the x-axis. And you can see that at the beginning of their lives, up until day 10, and then until about day 10, all of your worms are alive. And then worms, in this case at different temperatures, will start dying at different rates. And so you can see here at 15 degrees, it takes a lot longer for them to begin dying, up until about 20 days. And then the average, our mean lifespan, is calculated by looking at the number of days when there are 50% of the worms still alive. And so the mean lifespan of an adult that's been raised at a higher temperature than normal, or 25 degrees, is about 18 days. The mean lifespan of an adult worm raised at 20 degrees is about, say, 23 days. And at 15 degrees, it's like 27 days. And so ultimately, you will conduct a lifespan assay and collect your data, and then make a graph that looks very similar to this, and also interpret that graph, right? And so it's just important to remember that the mean lifespan is just calculated by looking at how old the worm is, or how old the worms are when there are 50% of them surviving. And so 50% survival, go down, and that is how you see your mean lifespan. And this is sort of an interesting experiment that comes from Cell Reports a couple years ago, and it shows that if you change the temperature, you can change the lifespan of the worm, but in different ways. And so if you shift adult worms to different temperatures, the effect is totally different than if you do it when they're larvae. And so that's sort of just one also interesting aside about how being exposed to different environmental factors at different stages of your life can actually lead to totally different effects on lifespan as well.